this episode of the Canadian Transfusion Trials Group Research Methodology Lecture Series. And we are kicking the calendar year off with Dr. Ryan Zarychanski. And for those who have not had the pleasure, Ryan is a professor of medicine, a hematologist, and a critical care physician at the University of Manitoba and Cancer Care Manitoba. And his research focuses on the hematologic aspects of critical illness and leads several national and international randomized trials in sepsis immunomodulation, anticoagulation, and blood conservation. And building on methods developed during the COVID-19 pandemic, Dr. Zarychanski and a growing network of investigators are advancing the principles of a learning health system by embedding clinical trials directly into patient care. And to improve access and reduce costs of clinical trials, Dr. Zarychanski and teams employ a variety of novel trial designs and research methods beyond the classical randomized controlled trial. And so this is, in fact, going to be the topic of today's discussion. I'll just mention as well, Ryan is the co-chair of GUARD, which is the Global Unified Adaptive Research Platform Collaborative, which is evaluating therapies for respiratory infections across more than 80 Canadian and international sites, including Brazil, Colombia, and Mexico. He provides leadership to several other novel randomized adaptive platform trials across a continuum of patient care. And in the transfusion and conservation space, he is the chair of the TRACTION trial, which evaluates the role of tranexamic acid in major non-cardiac surgery, and is the co-principal investigator of the PLEXUS trial, which is evaluating plasma exchange in septic shock. So it is our great privilege to have Ryan speaking to us today. And with that, Ryan, I will turn it over to you.
Thanks, Jacob. It's not that great of a privilege, but I'm glad to be here, and I think thanks for putting on this series, Jacob, and your leadership there. I did talk about platform trials last year, and some of this is going to be like some of it will repeat because I don't know who is all there, but also like as I was doing this, I realized that I couldn't stop like talking about just refreshing the RCT, and platform trials is just part of that. So. I think the talk morphed into a combination of like refreshing the RCT and my thoughts on that, but also a little bit more about platform trials and how I approach them and like the architecture. I think that this could be what other people want to hear about too, to some degree. And so I'm going to try to leave room for, discussion and questions. And then if there's like a big major topic that we want to come back to, then maybe we can plan that. I'm already volunteering Rob Belshaw since I see him on the talk and myself perhaps or anyone else interested to come back and give like a lecture on like Bayesian statistics for the clinician investigator, like as an example, because that features heavily into adaptive platform trials, and it's something that feels like could be further explored, like as an example. And so whatever like is mentioned today doesn't mean that it has to be the end all and be all, and maybe it's repetition and maybe it's whetting one's appetite. Okay, so I'm going to share something here. Oh, I don't want to share that one. Hold on. I want to share the other screen. I'm allowing something. I want to share it. I want to share this one. Okay. So I feel that you could probably see the slides now in present or some format.
That
If you can't, let me know.
looks good.
Okay. Okay. Oh, the disclosures related to trials, and a lot of them, I put these there at the bottom because these are almost all of them are adaptive platform trials now. Like Milocan. We'll talk more about Remap Cap, Clear Horizons, Brain Apt, and these are like platform trials, like you know from outpatients, Milocan, to inpatients ward, which is Guard, and from inpatients ICU, like Brain Apt and others. And so these really, like I say this because like they're part of what we're doing now because of the advantages, like that is embedding them into clinical care about the perpetual nature of them. It doesn't make them easy at all. And that's like one of the like take homes is that these are challenging, and to have a sustainability plan for each of these trials is immensely complicated, but it's important and it's possible. And you don't have to do it yourself, and you can leverage the existing platforms largely because we're building them purposely across a continuum of care. So you can like just do it, like reach out to one of the platforms and be part of it, or embed your stuff into it, and that's purposeful. To be honest, is what we're trying to do across Canada. Okay, so I'm going to highlight some of like the barriers to the design of like current clinical trials, and then introduce some contemporary methods. Talk a little bit more about Bayesian statistical frameworks and why, and a little bit more about adaptive platform trials and how we like prepare for them and set them up, and then some of the advantages and disadvantages. Okay, so some of this I talked about last time, but like the problem is like the RCT that we developed from the 1950s, we didn't develop it, but we inherited it, is old and largely unchanged. And like I dare you to think of one medical disease that we approach in the diagnosis and treat the same in 2025 that we did in 1950. And and so like my point is like medicine has evolved immensely. Like nothing resembles like how we approach medicine. Like very little approach resembles anything that was done in the 1950s or 60s from medical textbooks. But the RCT somehow survived until now. Like unadulterated, like unchanged in almost all of its forms, like even the method of consent, like and like the concept of randomization persists for sure, but like almost everything else, it's where we are now feels like not only antiquated, but it feels like it's almost inappropriate at some level because like the tool that was created, even though it did work for what we were doing back then, which was a lot of low hanging fruit, like does this like drug, does like Plex work in TTP? Like that's not a stretch, like you need like 100 people to show that. And the RCT it worked for that method, but the standard RCT and the outcomes chosen there is harder to employ now with the complex conditions that we're treating, our like our sort of goals to get at individual treatment effects, having biomarkers be part of this, etc. And so like even the tool may not scale well to where we are in 2025, but beyond that, like the evolution of medicine is what we sort of see and work within, but the evolution of the research design has lagged well behind that, and so it's time to refresh a little bit. Okay, and these RCTs are expensive and they take a lot of time to conduct, and I think that's really really important. Like this slide is that like 95% of the trials that we do now are underpowered, and they're underpowered by design because the methods that were handed to us are like prone to error. I'm not gonna say they're flawed, but they're prone to this error. And what it really boils down to is like a ton, like a sea of negative trials, some of which should be legitimately negative, but likely many of which are negative because they're underpowered to detect a binomial outcome, which is like then the binomial outcome is really, I may say that tongue in cheek, but it's like, is it positive or is it negative? So there's two things going on there. One is that we dichotomize the results of complicated trials into is it significant or not significant? And the New England like perpetuates some of this, like this false orthodoxy of which we should be, subscribing to. But we all do, to be honest. I just single them out because for many reasons. But and so we shouldn't be saying if something is positive or negative when something like is clearly on a spectrum of goodness or badness, and life isn't black and white. Um, but in addition to that, we are systematically misengineering the trials based on the framework, and largely that's the frequentist framework, chasing p-values. Okay, so it's been a big problem, and these are underpowered, and so this is like goes on today, and like I'm spending time on this because I feel it's that important. It's like this is like from 2024, and so these are like large CV trials in the New England Journal of Medicine. Have we heard of that journal? Yes, JAMA and Lancet, of course. And 82% of these trials overestimated treatment effects, 61 overestimated control event rates. And what will happen when you do this? Like, does anyone want to comment? Like, feel free. It doesn't, but you're likely going to say, well, you're at risk of underpowering a trial. Yeah. And it goes on. And it's like, it's so something from the past, something from the present, but still we don't believe it. And so I'm going to, not to single people out, but I'm going to single our Canadian trials out. And so this was a fantastic trial done by Deborah Siegel, and others that you'll mention, Emily Billy Gote. That was like a simple intervention that is small volume tubes to reduce transfusion. This is a question that I always wanted to do. I was shot down for many years, like the idea of it. And Deborah like had the fortitude and the leadership to make it happen. I'm like so proud of her. And she did this trial, and it was like heartbreaking confidence limits where the trial clearly reduced the need for transfusion, but it wasn't quite statistically significant. So it brought in like several things. One is that the dichotomization of results about like it works or it doesn't. And so I guess this doesn't work. But the second thing is like this was underpowered by design because of the framework we subscribe to, like these fixed parameter trials where we guess the control event rate, always wrong, mostly wrong, guess the effect rate, almost always wrong, the variance we don't pay enough attention to or nor simulate, and this is the result. Jessica Spence had the exact same outcome like just recently, like a year ago, we was studying benzodiazepines in cardiac anesthesia, and these like heartbreaking confidence limits that cross the null, interpreted as like in the abstract is like, can't see a treatment effect, even though there clearly is one. And it was underpowered by design and given a dichotomous like, treatment of, you know, work or not work, there's like no in between. Doesn't stop there. Like Shane English, Ottawa, like last year, New England, and like these like confidence limits that cross one on transfusion trials now. A second one, like another, the first one was transfusion two. This is different transfusion triggers in aneurysmal bleeding, like subarachnoid hemorrhage, and it suffered the same problem. Okay, like, and the Dean Ferguson, Alexi Turgeon, like again. So my point is, and this is the same thing. Like, I firmly believe, and we know that a higher transfusion trigger in traumatic brain injury is beneficial. Repeated in another trial that was like quote negative altogether in a meta-analysis. They're significant, but this whole thing is like crazy. It's crazy that I'm even saying this, like that this is interpreted as negative, and they are designed to fail. And we keep doing the same thing. And it's like, what drug, if you did this like 80% of the time, if you gave a drug 80% of the time and you knew that it wouldn't be beneficial or that it had high risk of failing and was, this was the outcome of wasting resources, and money, would this be approved and used? Like the answer is no. It would be tweaked. It'd be figured out like how do we make it happen better? And so we're here and like we're staring against a tool that is not working for us, and we've been slow to change. In addition, because of this, less than 5% of patients are enrolled in clinical trials at all, for which is needed to advance medicine. This research sort of enterprise is separated totally from clinical care. And we're missing the information, and we need the most informed practice. And so how do we go from this like random care we give and poorly designed trials to something that's randomized and efficient? And I think it's time to refresh the RCT. This is like a perspective we need to write. And so we learned a lot from the pandemic. It catalyzed the whole dialogue on how to reframe sort of like clinical trials, like where the importance of them and how they're placed into practice and how they're designed. And couched as an essential component of the knowledge to action cycle is probably where we need to be, a foundational element of the healthcare system, where good clinical care is represented by our ability to embed clinical trials into that. And so post pandemic, I think we're on to the fact that randomized trials from the 50s aren't the ones that we should be doing. And doesn't mean we have to abandon it, but we have to choose our tools a bit more carefully and choose the right trial design based, on the problem we're trying to fix. And the risk of the intervention, the outcomes, the consent model, etc. And there's different ways that we can do that. and these are some of them that I'm going to touch upon somewhat, but mostly focus on the notions of adaptive platform trials. And so including patients and the public and our research methods to design and conduct trials in a way that makes that is relevant to the stakeholders of which the patients are part of. So are we, using risk adapted methods of consent, considering adaptive platform trials a little bit more, employing Bayesian frameworks to a larger extent than we have in the past, and we'll talk about that a little bit more. Getting at that individual treatment effect. Right now, clinical trials return an average treatment effect, which is rarely informative to the patient in front of us. And so our holy grail is to get to the individual treatment effect. It'll be hard to get there, but we need to like pick up that torch and get there, and probably identifying heterogeneous treatment effects, or HTE, is a good start prospectively, something we haven't done before in clinical trials. I think the H trial is a way to expand our knowledge, develop biomarkers, um, hunt for HTE through nested translational studies, and could probably make better use of that. There are tools now that can enable broad collaboration, that being the multi-platform trial that we developed in the pandemic. And also, like, probably the largest single most method to decrease costs and efficiency and reduce error in randomized trial is to develop and make use of electronic data ascertainment where possible. And that's something that we need to move towards. And I'll give you an example of that. And so just quickly about patient public engagement. There's many ways to involve these stakeholders in trial design and conduct, from identifying priorities and research questions, selecting study outcomes, and creating materials and dissemination. We do this routinely in the trials that we conduct now. And one of the examples that I talked about before was Traction, where like I'm really like, I'm still really happy, and really satisfied that it was patients who developed the outcomes in this trial that were different than the one that I wanted to forward our study group. And this is a trial of 8,300 patients of tranexamic acid and non-cardiac surgery that's going to be hopefully published very soon. And so it was a co-primary contingent co-primary that was selected by patient stakeholders. And so by doing this, we increase the relevancy and impact of our trial. We ensure that the outcomes are really make a difference to not just investigators, to stakeholders. But there's advantages and disadvantages, or challenges as well, and it requires effort and relationship building, but it's really important. So as part of the sort of knowledge to action cycle. Our methods of consent, like, this is a whole talk that I'm not going to give, but they're antiquated, they're poorly understood, they serve institutions more than patients, and we're on to this. And so we're trying to reduce the consent model from like 30 pages to 15 to 8, and now we're getting down to approaching 5 or 6, using video methods of consent, risk adapted consent, etc. And different methods of consent exist. And I would encourage you to pick the right method of consent that matches your trial, and that are in keeping with the values and preferences, and and informed by patient and partner stakeholders as well. Particularly, we should be using risk adapted consent, waived consent, altered consent, and low risk trials, that are drugs that are licensed, that we know the risks and benefits of. Transfusion fits into this quite a bit. We did this in Traction. We used a really bespoke method of consent in Manitoba, and a total waived consent method, which was actually the request of the REB in Ontario. And these are other examples of that you might hear about, or that you could have heard about already. The trial that Michael Chazet led around donor sex and recipient mortality, and the REACT trials that are oncology, that use sort of different methods of consent, opt-out in this one, and oral charted consent in the REACT series. Okay, so let's just switch a little bit to platforms. And so, as I mentioned before, platforms are a different way of doing trials. And so platforms can be used two different ways. One is like platforms of research, which is sort of like, integrates networks and different trials, different trial designs of different stages under a theme, usually like stroke, to develop sort of an ecosystem research. Those like research platforms. Those are like different than platform trials, and these terms are different. And I just encourage you to pay attention to what people are trying to do and express yourself of what you're trying to achieve. Because some people have contacted me to help them build platform trials, but really they're interested in building ecosystems of research or research platforms and how one might integrate a whole sort of bunch of ways to do research through observational methods or registries. Or translational collaborations and move towards trials eventually. And that's different. That's super important, but different than platform trials. So typically we did trials, we conducted them or designed them, conducted them, analyzed them, and did the whole thing, over and over again, and built up all the infrastructure and took it down for each trial. And a different way to do that is under the context of a master protocol, have broad entry criteria into a trial or a goal of the trial, and develop an architecture to test multiple interventions at once or sequentially or over time. Learn from the trial and the data as it accumulates, make changes along the way. Like that changes could be like stop enrolling into something, start enrolling into something, change the allocation of enrollment, etc. And so those are two things here. One is the concept of the platform trial, which runs under the auspices of a master protocol. So, and again, you have a master protocol that kind of just says like what you're doing and gives like the ground rules for how you're going to do it. It might talk about the inclusion exclusion criteria. It might talk about the statistical methods, the governance, the major outcomes of the trial that may or may not relate to things you study in domains. But it's like it's the rules, and that's the master protocol. It mostly doesn't talk about exactly what you're going to study. It talks about how and in whom, in generalities. But then under the master protocol could have appendices or domains, as they're often called, where the actual treatments are that you will study are articulated much more completely. This is like the concept of the platform trial. That's different and totally separate than the adaptations that occur in platform trials. And so adaptations, which are on the screen, have nothing to do with platform trials, but platform trials lend themselves easily to adaptations, particularly in a Bayesian framework, because Bayesian statistics is naturally sequential and you learn from them as data accumulates. And so you can make changes based on accumulating data that fits naturally within that kind of statistical framework that's updated, with accumulating data. And so separate that out. So you can do adaptations in a frequentist framework. It's harder, less transparent, guided, like sometimes, um, not inappropriately, but they're challenged by the fixed parameters that have been sort of guessed at, to initially launch the trial in the first place. And so using a frequentist framework is a little bit easier to guide adaptations, but you can still do it in a frequentist framework. What are some of these adaptations on screen? And so you'll hear about them. Again, they're separate than the framework, separate than the platform trials. And I just want to just make that very clear to some of the earlier learners or people who may not have had exposure to that. Adaptations are pretty groovy and they can be really powerful, and they're used to increase power, to increase efficiency, sometimes of enrollment, sometimes to reduce like um, wastage of resources or to improve like the cost efficiency of trials. And so I'll give you some examples. And so like the first one is response adaptive randomization, which gets talked about a lot, more than it deserves, to be honest. Like, and what this is that, and there's an image beside it where you change the randomization allocation. So usually it's a coin toss, 50-50, right? People get randomized to a drug or not. And it's usually in a balanced randomization. And people like mostly are okay with that, but it is not the way we like practice in medicine, like often in medicine, if we have a notion of something that's working, we kind of want more people to get the drug that's working. That's like us providing care as healthcare professionals. We want to limit risk to people who aren't benefiting. We also, it doesn't respect the fact of like learning. Like, as I said, like, Bayesian statistics are naturally sequential and can learn from accumulating data. And response adaptive randomization, like it incorporates these notions where the trial can learn about what's working and change the randomization allocation based on what's working. And so more people get randomized to the drug that's working and less people get randomized to the drug that isn't working. And so it's sort of like is a very ethically comfortable way to run a trial. And we've used that a few times in the trials we've done. We've got it right a couple times, and one time we got it really wrong. And so ways that we got it wrong, which we should have known about, to be honest, and it was like a bonehead rookie thing we did like many years ago, where RAR is not a good idea when you only have two drugs that you're studying. Like it really works when you have more than two drugs because it doesn't increase the power, and in fact, it can reduce the power, but it might increase the ethical acceptability of the trial nonetheless. And then you have to figure out how to change this balance ratio. If you change it too much for drugs that are working, then you could inadvertently unblind the trial if people starting to get randomized to the same intervention, obviously. And I did make that mistake once. Okay, so sample size reassessment, like this is like a big deal. Like, as I said, most trials are underpowered by design, and we make guesses wrongly, and we can reassess the sample size iteratively and like, over time as data accumulates so that we can take the next step of adaptation, which is stop the trial when you have a result, as opposed to like, when you've reached your sample size based on guesses. Right now, that's what we do. We stop our sample size based on guesses about the effect size, about the baseline event rate, which I said we got wrong, and I showed that. And another way to do that is to stop when a trigger is met for superiority or inferiority or or futility. And so these can be all be sorted out a priori. There are other designs. One of them that I think is powerful, these seamless designs, like phase two, phase three designs, where something might be really expensive or risky, or you're not sure you want to go all in on a phase three. We have a design that we've used a couple times now where we have graduation rules. We start something, we say, okay, well, let's like, here's a company that will, here's an example. It doesn't have to be like this, but a company that will pay for the drug for the, first few hundred patients. So we're going to have a graduation rule for the trial that we need like some probability of some treatment effect to go further. And if we don't see that, then it's likely, not going to be feasible, or just won't be acceptable if you don't see this kind of treatment effect in practice, given the side effects or costs. And I think this is a way that we could, improve the efficiency of investment and not take everything to a phase three when at a graduation phase that makes sense, or some decision rule phase, it's like the treatment effect is like too small, or the costs are too great for the treatment effect or something like that, or the side effects or something is like the risk is too great to go further. And we don't make enough use of that as well. In the notion of like looking for HT or heterogeneous treatment effects, like I think we can and should make greater use of enrichment designs and include it into that or de-enrichment designs too. And so like as the trial progresses, we come across an average treatment effect. Of course, that doesn't apply to all patients. And so if we can figure out populations by biomarkers or outcomes or something, if we can discern populations that may be benefiting or not benefiting in trials as data accumulates, we can choose to enroll more of those patients or less of those patients so that we can improve the statistical efficiency and impact of the design over time. Okay. And so the end result is that in a platform under a master protocol, in a platform trial, that is, that you can have a group of patients start multiple interventions, evaluating them, and that is, you may stop intervention when it meets a pre-specified outcome for whatever, in this case, futility. You might add another drug, that at that time for based on health priority or new evidence that's emerged, and then continue the trial in a seamless fashion. And so this is like the notion of platform trials, and you can embed these into clinical care. This was done really well in the pandemic with remap cap, where instead of studying, A and then B and then C, all of those things, A, B, and C, were studied at once. Over time, allows you to compare the drugs to each other at the same time and even test for interactions as they occur. Remap Cap did this really well. They've randomized probably over 20,000 patients now and like many hundreds of sites and high impact publications. And it wasn't just because of COVID. Because people say, well, that was COVID. It was easy. It wasn't just because of COVID. It was because the trial fundamentally came up with an answer. And that answer was important. The answer was it worked, it didn't work, or this is not worth studying further. Like, which is another way of saying not working. Like, it's like we've done like so many patients, you're never going to find this answer in a feasible method. And each of these are like reasonable answers. And that's why they're one of some of the reasons why they're in high impact journals. So this has taken off a lot since the pandemic. And these are some of the trials that persist or have been developed. So I help chair a trial called GUARD RESP. And GUARD is the Global Unified Adaptive Research Platform in acute care diseases. And so GUARD RESP just studies respiratory infection of that. And it focuses on non-critically ill patients, where REMAP CAP focuses on critically ill patients. And so we're studying different interventions in pneumonia, but Todd, Lee, and Emily are starting a whole different domain in another infection, respiratory infection, where I'm hoping to work with CanVector to start GUARD VTE, where we can study VTE in patients. Patrick Lawler leads a post-sepsis or post-discharge platform called Clear Horizons. One of my mentees, who's a faculty now, Brett Houston, leads like the first of its kind adaptive platform trial in myelodysplastic syndrome and low intensity AML, largely an outpatient trial, evaluating different therapies in a high risk population, high risk for death, across Canada, and hopefully to develop international collaboration soon. Alexi Turgeon runs Brain Apt, which is an adaptive platform trial that's embedded into clinical care, studying different interventions in traumatic brain injury. Ewan Gallagher leads Practical, studies hypoxemic breast failure in the ICU, and there's multiple other ones. And so there's a bit of a tsunami. These are the ones that I think that have legs that will like persist. I think Donnie and Francois Lamontagne with the trials group have started a platform to study different supportive care interventions in the ICU, and I think that that's going to have a lot of potential as well. And so it shows the investment that others are making. Talk about GUARD a little bit more, just to get a sense for the architecture. And so GUARD is that Global Unified Adaptive Research Platform studying respiratory infections. So GUARD RESP. And so the statistical model studies a primary outcome of organ support free days. That's it's not that important what it is. It's trichotomized, so it's over like I'm not going to say what it is, but it's a primary outcome that's appropriate for the patient population. And there is a master protocol, and their master protocol is like a document that's super boring. And it really talks about like kind of what the goals of what we're trying to do in life with this platform trial, and the outcomes to the trial that are important. They may not relate to every domain, but they mostly do. So the patient population we're interested in, and the governance of the trial, like the committees and the statistical like generalities for how we're going to like approach the trial with regard to simulation and analysis and adaptations. But all of that is fleshed out further within each domain. These are the domains. And so, sorry, I didn't mention the syndrome. So the syndrome of CAP is one that we'll focus on here, but we have another syndrome that is PJP now, and another syndrome that's being under development, which is fungal. And so you can see like this is like a platform in its own right, but the platform trial is each of these syndromes. And so under CAP, we have different domains of anticoagulation, we're studying heparin. Another um, domain studying corticosteroids, which is called DefenCap, the intervention there. We have another domain of antibiotics. We have a prospective registry that is non-randomized, and others will come. And so you can see like that we are studying multiple things in pneumonia, high risk, high prevalence this disease, most common cause of hospital admission. And we're doing this like I think there's 60 activated sites now in several countries. We're expanding to different South American countries and Australia in the next year and New Zealand. I think we have about 700 people enrolled so far. And this is just in the anticoagulation one, and this will take off further once the steroid one starts to enroll. We're studying steroids, we're studying antibiotics, and and the antibiotic one so far is a pilot. So this is an embedded pilot within an adaptive randomized platform trial. This is the design of Brain Apt, where people get randomized, and this is just a way of expressing what we're doing in a platform trial. And there's planned. I don't like the word interim. I like the word adaptive analyses. This is a slide from the trial itself, but I like the word adaptive analyses because um, I think that the decisions are are made based on the data that accumulates. That isn't interim. It's because that implies like when the trial stops, people start using words like early stopping, which has a bad connotation, but it's not early stopping. It's like appropriate stopping based on a prior decision rules. And so that's why I don't like to use. I think adaptive analyses is perhaps more suited when talking about adaptive platform trials. And if a conclusion is met, then great. Then you like disseminate the results. And if it's not, then you continue randomization and continue adaptive analyses at a priori like defined times that are like not free. You still have to have type 1 error control in this until a conclusion is met. Milocan does something similar to this, where there's a master protocol broken into two severity states, intensive and less intensive. And then there's different sub protocols or domains, as I call them in my trials. And studying tranexamic acid is one of them, an antibiotic one is starting. And, just to give you examples of these in Canada. Okay, not a Bayesian lecture, but I want to stress that, this is like a statistical framework that is like, you know, that has been not new. Um, and should be more used. To be honest, it's easier to interpret than the results of frequentist frameworks. We're used to the frequentist framework because we've been taught it in medical school and grad schools. Um, um, and it has major disadvantages and likely probably should be retired, probably. I mean, that feels a bit controversial and polarizing. It's not meant to be at all, but like there's just not a lot of obvious advantages when one considers other alternatives, include like to the Bayesian framework. And so the Bayesian framework sort of like it's akin to how we think, like where we and how we diagnose in medicine. Like it's so funny when you have a lecturer giving a diagnostic talk, they will talk all about pretest probability and likelihood ratios and post-test probability. But when we talk about therapies, we only talk about does it work or not. And it's so artificial because like nothing like works or not. Like you're not like I'm not mad or not mad. I'm just thinking about like how I get parent for some reason. But like I'm like slightly annoyed with you right now, my child, or I'm really frustrated with you, or I'm furious. Like maybe it was a reflection of how school started this morning. But we dichotomize the treatment effects, but we naturally understand the notion of probability when we're making the diagnosis, which is like just this is cognitive dissidence. Like this is like a rut that we're in that we need to get out. And so we can apply these same things to treatment effects as well and get and evaluate what we think on more of a spectrum of probability. This is how we think already. And Bayesian statistics offer a lot of advantages. Like, they're easier to, plan and affect adaptations in adaptive platform trials. That's for sure. But fundamentally, it gives us the answer we want. Like, what do we want, Donnie? What do we want from a trial? We want to know if something works, right, Johnny? Like, we want to know, does something work? And we also want to know like how well that it works. How confident are we that it works? And that's exactly what a Bayesian trial tells us with the credible intervals. It tells us the plausible range of treatment effects and the probability of seeing a certain effect. The probability of seeing a certain effect. And it avoids this dichotomization as something that works or not works, or and ignoring it if there's still some treatment effect that didn't reach our like threshold of statistically significant. It tests what we want to know, the probability of treatment effect, rather than what we do in a frequentist framework, which is the probability of theoretically, because it's long run statistics that we don't do the long run of. So it's a probability of theoretically observing an effect that's greater or equal to than the observed effect, assuming that there is a no effect in the trial if it was repeated in a similar population. Do we go to a patient's bedside and explain that result of the trials we do? No. We tell them if the probability that the treatment works. So we automatically Bayesianize the results of a frequentist trial because that's already the way we think. We don't even understand the results of what that I just said, and we don't apply that at the bedside. It should really twig us to the fact that like the framework isn't fitting even how we want to talk to each other and to our patients. It allows the trial to be conducted without making guesses about the event rates or the treatment effect. You can still make guesses, and I encourage you to if you have like knowledge in the field. But when you don't know, you can still do the trial. You'll have to simulate what the cost of not knowing is, like having very neutral priors, assuming like conservative treatment effects, etc. But it avoids us locking in the parameters at least, and we can still update the parameters based on data that accumulates. Like it's everything we want, and the benefit here is that reduces underpowered trials, improves impact. It allows the adaptations to be incorporated as data accumulates. I mentioned this already. Um, and it's the way we think. I mean, it's the way we diagnose. Here's just an example of, D-dimer and like pretest probability. Why isn't treatment a probability as well? It is a probability. We just ignore that. And it's easy to interpret. Like, people like somehow get a little bit freaked out when they see like posterior probability or whatever like this. Like, well, let me just tell you that like this is an odds ratio of one or greater than one. So that means any treatment effect that's beneficial in this case, any treatment effect. So if I tell you that there's a 98.6% chance that there's a treatment effect that's beneficial, how beneficial do you think that is? I think most people would say that's likely beneficial. Who cares what we're talking about? Well, I think we should care because does that treatment have side effects and what are they? Does it have costs and what are they? Acceptability. But we can tell you the treatment effect with some degree of certainty as well. We could change this too. It's like, well, this drug is expensive and I need to see an odds ratio of 2 that translates into an absolute risk difference of 5%. I'm making this up, but like because a bit depends on the incidence of the in the population, of course. But let's say that was the case, and so then you could just put this into an app and say, hey, what's the probability of a treatment effect of 2 now or 1.5 or something like that? Because this drug is expensive, or this drug has a side effect that I wouldn't tolerate or accept unless I had this kind of treatment effect. And this is easy. You can just put this in an app, a Shiny app, and tell you about this. Try to do that with your frequentist trial. And so like there's just so much more at your disposal with this. And I'm not like some evangelist here. I'm just like letting you know that what I think about it and how I've incorporated this into the trials designs that I try to get involved in. The simulation is everything. And so the Bayesian trials like have this ability to understand all of this, like to the extent that we can before a trial is launched. And we do maybe a little bit about this conditional power or something with frequentist frameworks. So I bet you most people haven't done more than that. But like you can really dig in, like take a deep dive using Bayesian statistics into like understanding that the trial power, that type one error, the range of sample sizes you'll need. You can explore how this performs under different control event rates and then design your adaptations, with with the statistics in mind. Look for, subgroup treatment effects and HTE. And I give you some examples of that. This is a design report for that I do for every trial that I get involved in now. So this is a design report for a TAP. Um, Let me know if you can't see this, but I think you can. And we do this with our statisticians. We work as a team. We talk about the model that we're going to use, yada yada, and some of the assumptions and what kind of priors we're going to use. And then we get to like, okay, well, and these are the outcomes. This is like the outcomes. Like, so what's our definition of superiority? What's our definition of futility? Okay, and so it's under some assumptions and control event rates that might vary. Control event rates that might vary. These are the high risk and a low risk group, and the odds ratios that we could see. Like, this is all simulation under all these parameters. What do we see? And so we can say, listen, if the trial, what is it showing here? Hold on. This shows the trial doesn't work. So let's say like the trial has no effect. This is no effect in here. Well, let's make sure our type 1 error control is good. So type 1 error control is good. Now let's say the trial where it's in a low risk and a high risk group. Let's say there's an odd ratio of 2. What would our power be and our sample size at that power? We can say, well, okay, well, now we have 80% power. This be like 700 patients. Trial didn't work as well. 1.35 odds ratio. We got 80% power. Like, so that's just like an easy example. I'm really looking at type 1 error control here, but it gives you a reflection of what kind of sample size you can use. Well, then simulate further. Well, let's say that in a high risk group and a low risk group, the treatment effect is different. And these are the treatment effects. Well, now what will I be in for each group with regard to power and sample size? Um, you know, and so we go through this. This is just a power calculation, but we can do this for all of the trial. We can incorporate statistical borrowing, where you say, well, the subgroups are the same and they get combined. How will that affect our ability to make a treatment decision, etc. And so I just want to get the impression across that in this framework, all of this is simulated to the extent we can to understand how our trial parameters and guesses work out under different assumptions. And this is a super important part of designing a trial in a Bayesian framework. Really under any framework, but it's just a bit easier with the Bayesian framework. And this allows us to look for heterogeneous treatment effects. And so, as I said, like most trials, well, all trials give an average treatment effect. We look at subgroups that, rarely impact our practice and usually herald the need to trial something again. But I really want to get to the fact that, I gave this example in the last time I provided this talk, was that we are now experimenting with prospective methods to detect different treatment groups in identified in-trial subgroups. And so this was an example in the pandemic where we broke the patient population into severe and moderate. We had statistical borrowing that could occur between the groups if the treatment effect was the same. And we could report them out separately if they were different. It also shows how we used response adaptive randomization, like here. This is what the blue is, to change the allocation ratios based on groups that were benefiting or not benefiting, and allows us to come with different conclusions based on the treatment effect in different groups. Now, that's in the past, but it still was pretty practice changing, like our trial methods changing, like for us in our population, not in like maybe some other like groups and like weather prediction and other like robotics or whatever. And so in GUARD, we're doing this as well. So we have a 2-stage design where the trial starts, and at 1,000 patients, we have an adaptive enrichment analysis based on different prospective variables that we've identified, informed by the previous trials that we did about BMI and CRP and pneumonia severity index. That's the PSI. And we simulate all this. And so this can be complicated, but really I just want to break it down into like my interpretation for you. So in the top left here is like the odds ratio. So the odds ratio in low PSI, high PSI, or high CRP, low CRP, in any group, it's one. It doesn't work. And so at our adaptive analysis, we have a 41% probability of continuing the trial versus stopping, which is good. It was like if a trial doesn't work, why would we go to like the very end of of our calculation? So this allows us to stop, you know, halfway through, which was like 3,000 was the maximum sample size for the effect that we could see plausibly. Likewise, if the trial worked in everyone at our adaptive enrichment step, we have a 90%, it worked 1.5. That's 1.15, the odds ratio, 1.15. In our adaptive design step, we have a 90% probability of continuing. Hey, there's a 10% chance we would quit, but we're willing to take that risk um, because of what we see if there's heterogeneity coming up. And so we have a sample size that's not very large now, 1,500 mostly, um, if it because we're looking for these differences, and we didn't make much of a change here, by the way, because it was working in everyone. But let's say it's working in one group, the low PSI, but not in the high PSI. Well, now with our a priori rules and our simulations that we've done, these are all simulations, but these are like the best, not best case scenario, like the most plausible that we've come up with, is that in the group that's benefiting, there's a very high chance that will continue in the trial. And in the group that is not benefiting, there's a 50% chance we'll stop. That doesn't sound great because it's not, but it's 50% better than we would normally do. Like if we didn't look for this, which we normally don't do, we do subgroups after, we'd never find it. And so now we are looking for it, and our tools only have discriminative power to look to find it like half the time. So it's not perfect. These are evolving methods. This is the best we can do, but I'm just showing you that we're building in a priori tools to detect heterogeneity as trials progress. Okay, I'm not going to talk more about like the multi-platform trial, but it's ways to spur on global collaboration using methods that we're going to be publishing a paper on very soon. I will just mention electronic data ascertainment, which I think is like the biggest thing that needs to come right now. In the traction trial, we, did this 8,400 patient trial for like $1.8 million. It's because we didn't collect any data. And like an analogous trial like POISE cost $10 million. And so you can do large trials like Laura Lynn's fluid trial, Nancy's like informed trial, huge trials for like very cost effectively if you're not collecting data that's already collected. And so that's the next place we need to go together as a country, as really as an international consortia. Okay, so I talked about like refreshing the RCT, about all these things, patient public engagement, and a bit about consent, more on adaptive platforms and Bayesian frameworks. And somehow how we're using methods to detect HT, and they're not perfect, but we're starting to. And just editorial comment on data ascertainment. That could be electronic. This is an example I gave to the PCAN network about how they might design an adaptive trial to meet the needs for people with pulmonary embolus in the hospital. I just made this up. It's just like it's theoretical, but it's like an example of where I kind of hope that I can take this group, with their help and engagement and enthusiasm and expertise, so we can have an inpatient platform trial to study pulmonary embolus, which I think is something that hasn't been well studied in inpatients like VTE, like DVT has been. Okay, I think that's already enough. I won't talk about challenges because I could go on for a long time, and I'm out of time. And this is like the goal here is to embed these into clinical practice. So then we're caring for patients as we're learning to care for patients at the same time. Thanks, Jacob and others, and hope this was like somewhat useful, even though it was a potpourri.
Fantastic. I have all sorts of questions, but let me open it up to the audience because we don't have a
Yeah, and I
huge.
didn't leave a lot of time, and I apologize.
Yeah.
I have a quick question. Is that okay?
Donnie.
Yep.
Yeah. Hey, Ryan, thanks again. That was really
Hey,
great.
Donnie.
So, when we talk about things not evolving,
Yeah.
um, peer review committees at CIHR don't evolve very quickly, and
Yeah.
sometimes have a hard time getting their head around, let's say, adaptive designs where you actually don't know what the ultimate sample size is going to be.
Yeah.
Anyway, so quick question about, do you have any strategies about how to, articulate that? So that's number one. Quick number two is you've talked a lot about the stats kind of skill that's required for
Mhm.
this, which I think is so key. And if you have a good statistician or team of statisticians who understand this, you are so far ahead of the game. Is that a group that you have kind of leaned into and created, or how do you go about
Yeah.
sourcing those
For sure. We
people?
have like a good group now, and we're building expertise in Canada too. And we've got some training programs. We have some, again, we have like the senior groups that we like pay like $300 or $500 an hour for, which I don't recommend. And then we have like the only when you need to, and then we have the groups that are like more like on our side, and they're collaborators, and we have analysts in between and to fill in the gaps there. And so we are developing a national network, and we do have people that can work with individuals who want to bring this to the table. The funding is a problem. We're talking to CHR right now. Like we've had like discussions with the very like the head of like the institutes of the CHR about the need to fund platforms more and adaptable platforms. The sample size is an issue. And so we sort of like have been, our strategy so far has been sort of talk about feasibility of a maximum sample size and mitigation strategies and how this will be used, how the funding could be used in other ways, should we not get to that point. But it's an evolving conversation that's far from perfect, and you hit the nail on the head there with Donnie. It is like squarely, I might add. It's a challenge.
Thanks.
Yeah.
Rob, looks like you may have a question.
Comment and a question. The training of highly qualified statisticians who are not, I grew up in a frequentist school and learned clinical trials in pharma, and it's a slow change. It's a sea change in the way you think. Ryan, I wonder if you have any opinion on whether regulatory trials that are going for products or well products that are going for regulatory approval. Do you think that that's a distinction in the adaptive Bayesian space, or do you think that this stuff works everywhere?
Well, the NIH is accepting it now. I can't speak to regulatory like approval for new drugs in Canada. I don't know. Actually, like I don't have any experience with that.
Okay.
And I still have more experience in the States because I just have like some more colleagues that I collaborate with there, and so the NIH is now more comfortable with this. It's not seamless and it's perfect, and there's a lot more oversight and like suggestions by them that are like mandatory suggestions, which is different than in Canada where like you don't have that sort of influence, and so there's a lot of co-development that's going on there right now.
Yeah. I'll just mention this, the ICH, FDA, Health Canada, that organization, they're putting out guidance documents now, draft guidance documents on the use of Bayesian stats in trials. So I think you are leading a charge here.
Rob is a statistician, and he's like, part of this community, and we are evolving together. So thanks, Rob, for those things. And I always feel self-conscious when I talk about statistics in front of a senior statistician, but I hope most of it was correct.
Okay. Listen, we're just about out of time. But fantastic talk. As always, Ryan, thank you so much.
Well, thanks for putting up with it, and then thanks for being like, Bayesian curious at least, and we'll see where we go from there.
Absolutely. So yeah, for everyone else, do join us next month, when it's going to be Rob actually speaking about statistical considerations at greater length. And please do remember to fill out your evaluation form. And with that, we will adjourn.
Thanks, Ryan. See you later.
Thank you, guys. Good to see everyone.